This presentation was made for members of the Wasatch Front Water Quality Council to help them better understand the importance of developing food web models for Utah Lake. It is also general enough for others to view and be informed of our development of food web models for the lake. The big question, why are food web models critical for managing nutrients, fisheries, and other factors, including controlling cyanobacteria blooms? Short answer, basically because without these models we won't know how all the factors interact and which are limiting factors. 2, we won't be able to predict or evaluate any management outcomes. And 3, piecemeal attempts such as nutrient reduction alone to control blooms will likely be futile. We all know that nutrient reduction from water treatment facilities alone will not reduce algal blooms. Sediment flux and atmospheric deposition will continue. Equally as important as nutrients are the top-down controllers of algal blooms, i.e. the food web. Food web models are on the to-do list of Utah Lake Science Panel research agenda. As food web modelers, we like to state, organisms are not just chemicals. Personally, I like to state, organisms are not just chemicals, they are chemicals come to life. Contrary to popular dogma, cyanobacteria blooms and nutrients do not occur in a vacuum in Utah Lake. They are but a part of how Utah Lake's ecosystem functions, and other top-down factors may be as or more important to blooms than nutrients alone. Utah Lake's food web and ecosystem functioning is far out of balance for a variety of reasons, including natural conditions, but mostly management actions and society's past disregard for Utah Lake's health. Here is a simplified Utah Lake food web model. We are all familiar with nutrient and algal bloom relationships, including water treatment facilities, atmospheric deposition, and sediment resuspension. We are less familiar with top-down factors. As an analogy, we are all familiar with the idea that properly managed cattle grazing can improve range conditions and even increase the nutrient quality of the grasses that cattle feed on. Zooplankton are like cattle, or deer, or elk, or rabbits, or even bison. Zooplankton graze on phytoplankton and help determine which phytoplankton species occur, their nutrient quality, and their abundance at any given time. Zooplankton are the most direct top-down link to phytoplankton diversity and abundance and blooms in the lake. However, zooplankton are also the favorite food items for planktivorous fish, including carp at all life stages and every other juvenile fish species in the lake. In turn, planktivorous fish are eaten by piscivorous fish, that is, fish that like to eat other fish, such as the two most abundant piscivores, largemouth bass and walleye. Here is the basic food web of Utah Lake as food web modelers and ecologists see it. Starting with producers, the plants. Consumers ones are invertebrate grazers such as zooplankton, filterers, collectors, gatherers that feed on producers, the algae and the plants. Consumers 2 are invertebrate predators such as dragonflies and damselflies and juvenile fishes. Consumers 3 are fishes that feed on consumers 1 and 2 as well as producers. Producers are dependent on the chemistry of Utah Lake, particularly nitrogen, phosphorus, and temperature. The decomposers are mostly bacteria. All the groups interact and contribute to detritus. Of course, the food web is much more complicated than that illustrated. Here are the major players in the food web of Utah Lake illustrated in the previous slide. Take a few seconds to examine the many major players. Some of the basics that we are pretty much sure of. Phytoplankton assemblages, including cyanobacteria, vary spatially and temporally in the lake. The figure on the right is an example of some of our research, showing how phytoplankton assemblages follow a seasonal pattern. As you can see in the figure on the right, there is a clear seasonal change in phytoplankton assemblages with basically 4 groupings: February, March, and April in the left top quadrant, May, June, July in the bottom left quadrant, August in the bottom right quadrant when cyanobacteria blooms dominate, transitioning through September to October, November, and December assemblages in the upper right quadrant. Notice the change from December to February and early spring assemblages. We, along with Rushforth's phycology, have built one of the most comprehensive Utah Lake algal taxonomy and ecology sets available. Zooplankton assemblages also follow spatial and temporal patterns in response to and due to their grazing interactions with phytoplankton assemblages. NMS ordinations show these patterns extremely well. Notice the figure on the right that shows an almost circular pattern of seasonal changes in the zooplankton assemblage, mimicking the phytoplankton assemblage seasonal patterns shown in the preceding slide. We, along with River Continuum Concepts, one of our contractors, have the most comprehensive and up-to-date zooplankton taxonomy and ecology data sets for Utah Lake. We have also modeled interactions between phytoplankton and zooplankton assemblages in the lake. The figure on the left is a conceptual path analysis diagram of potential pathways between phytoplankton assemblages and zooplankton assemblages. The figure on the right is a structural equation model that verifies the path diagram. No need to go into the details here, other than phytoplankton assemblages and zooplankton assemblages indeed interact within Utah Lake. There is a mutualistic positive feedback loop occurring in the benthos in the lake. It is the mutualism between midge larvae and benthic algae. Midge larvae are by far the most dominant benthic invertebrate in Utah Lake as measured by density and biomass. Two species of midges dominate, Chironomus species and Tanipus neopunctipinus. Densities of these midge larvae typically approach 10 to 20,000 per square meter. Vast amounts of midge biomass are needed to support the Utah Lake fisheries that rely on this energetic food resources. This positive mutualistic feedback loop between midge larvae and benthic algae is essential to main productivity in the lake. Heron et al. 2017 showed that as midge larvae densities increase, algal production increases, and consequently larval growth rates increase as their densities increase, likely because of the increase in algae. Without this positive feedback loop, far fewer fish would be supported in the lake, and just as importantly, nutrient uptake by benthic algae would be substantially less. Midge larval tubes are easily observed in soft substrate in Utah Lake when the water is clear and shallow enough. Holes in the sediment are midge larval tubes shown in the three photos. Inside exclosure cages, absent of predators, larval tubes were uniformly distributed, suggesting intraspecific competition for space, i.e., midge larvae were at carrying capacity. Midge larvae tubes were less dense and less uniformly distributed outside of the exclosure cages, in this case in Farmington Bay. Shorebirds ravenously fed on larvae in Farmington Bay. In Utah Lake, it would be the hungry little fishies that keep midge larvae from reaching carrying capacity. The bottom image shows the three-dimensional structure of the tubes, which alter sediment chemistry, structure, and function, which we will discuss in the next slide. Here is another amazing and very important fact. Midge larvae can indirectly affect algal blooms. When the larvae are present, they oxygenate the top few centimeters of the sediments via activity pumping water through their tubes, which allows iron to bind with phosphorus. When midge larvae turn into adults and fly away, the larval tubes collapse, and top layers of sediments can turn anoxic, and iron releases phosphorus into the water column. Of course, there are much more chemistry and biotic interactions going on, but you get the picture. Without understanding and modeling the food web, we will never know how to control algal blooms. Starting in spring and early summer, zooplankton abundance and diversity skyrockets in hyperproductive Utah Lake. Here is a video of thousands of zooplankton, copepods, cladocerans, and rotifers heavily grazing on phytoplankton and keeping water clarity high in Provo Bay. Nutrients fuel this productivity. Without a diverse and abundant zooplankton assemblage, algal blooms would be substantially more frequent and intense. Shortly after zooplankton populations bloom, packs of carixids, water boatmen, arrive. These insects prey on zooplankton, benthic invertebrates, and detritus at incredible densities in the shallow areas of the lake. Carixids and zooplankton are major food resources for juvenile fish and minnows. Their role in the food web and indirect effects on algal blooms is unknown, but we intend to find out. Juvenile fish occur at very high densities, feeding on zooplankton and other pelagic and benthic invertebrates, particularly during summer months when almost all fish species have juveniles swimming around the lake. All juvenile fish in the lake, including piscivorous adult fish such as largemouth bass and walleye, feed on zooplankton, as do benthic feeding catfish, and our nemesis, the carp. Competitive release of zooplankton for removal of large carp is an example of unanticipated or unmeasured management actions. This is one of those important food web interactions that needs much more discussion and analyses if we are going to be able to manage algal blooms in the lake. As we showed in earlier slides, zooplankton are the #1 top-down controller of algal blooms within the water column. If there is an overabundance of planktivorous fish eating zooplankton, which we postulate there is, then the food web is out of balance and the zooplankton cannot help regulate the blooms. The next slide will help illustrate this. Here is a hypothetical but reasonable trophic level diagram of changes in trophic structure that appears to have happened in Utah Lake and what could happen if we conduct biomanipulation of the fish populations. These are some of the types of output our food models will generate. In this scenario, pre-settlement conditions had an abundance of top-level fish predators, the Bonneville cutthroat trout. There was also a high abundance of zooplankton and a low abundance of phytoplankton. Post-settlement to present conditions on the right side of the graph have fewer top-level fish predators, fewer zooplankton, and much greater phytoplankton. Biomanipulation of top-level fish predators could push conditions back towards pre-settlement conditions, although this would be an analog food web. We have started using EcoPath with EcoSim, abbreviated EWE, food web models that are the most widely used fisheries models worldwide. More than 500 research publications have been made using these models, and their number is rapidly increasing. EWE models are now being used to help managers and governments manage fisheries and aquatic ecosystems all over the world, modeling the effects of nutrients, global climate change, invasive species, environmental changes, and fisheries management strategies. Once we populate the EcoPath mass balance models, we can then use EcoSim and other add-ons to predict changes in the ecosystem and different management outcomes, including, of course, the one we are most interested in, nutrient concentrations and loads. These food web models will allow us to determine if nutrient reduction alone will reduce cyanobacteria blooms, or if more cost-effective and prudent management strategies can be implemented. We are coordinating our modeling efforts with DWR fisheries biologists. They are enthusiastically on board. In a nutshell, these models are a big reason why all the data on nutrients, chemistry, and biological data are being collected on Utah Lake. It is my understanding that DWQ has basically assumed, although they might not admit it, that Wasatch Front Water Quality Council and Oreohelix Ecological are the local experts on Utah Lake food webs, and that the council will likely take the lead on food web model development.